Hello and welcome back, AP Biology students. I hope you're all doing well. This is part 8 of our cellular energetics lessons. It's the second lesson focusing on photosynthesis, and you can see today we're going to cover pages 164 to 171 in the Campbell Biology in Focus textbook. So let's begin today by looking at the overall chemical equation that summarizes the photosynthetic process. And that's exactly what this does. This is just an overall summary of the chemicals that go into photosynthesis and the things that are produced by photosynthesis. There's two different versions of this equation that you may see in various biology textbooks. I prefer the one that I've highlighted here, the simplified version. I much prefer this one because it has the three reactants on the left, the two products of photosynthesis on the right. Yes, it's true, technically light is not a chemical reactant, but we're going to lump it together with carbon dioxide and water because all three of those things are required for photosynthesis to occur. And the two things that are produced by photosynthesis are glucose and oxygen. So let's take a closer look. Again, when you look at this simplified photosynthesis equation, the three items on the left, that's 6 molecules of carbon dioxide plus 6 molecules of water plus light. Those are the three reactants. And the products of photosynthesis are 1 molecule of glucose, C6H12O6 is the formula for glucose. Also 6 molecules of oxygen are produced, and this whole process is made possible by the green pigment chlorophyll, which must be present in order for photosynthesis to occur. Now it's easy to, when we look at this equation in isolation, it's easy to forget that this is something that plants have to pull off. Plants struggling to grow and survive in nature have to make this happen. And so it's important to think about it from the perspective of a plant. If you're an oak tree growing in the forest, how are you going to obtain these three necessities, these three reactants? And then what are you going to do with the products once they are produced? So let's look at it. I'm kind of zooming out now, looking at the equation as a whole, and let's break it down in terms of how plants actually pull this off. So the carbon dioxide, which is the source of carbon, which is necessary in order to make this 6-carbon sugar, that carbon dioxide, of course, is available in the air. Carbon dioxide is given off as a waste gas by animals and fungi and bacteria and protists, and even by plants. Plants produce carbon dioxide when they do aerobic cellular respiration, same as we do. So carbon dioxide is going to be available in the air surrounding the plant. But if you'll recall from our previous lesson, photosynthesis, at least plant photosynthesis, occurs in the middle of the leaf in an area called the mesophyll. So the only way that carbon dioxide can get all the way into the mesophyll is by passing through very tiny openings in the bottom of the leaf called stomata. For most plants, water is absorbed from the soil by the roots, and there are millions of microscopic tubes that travel from the roots up the stem to the leaves, and those microscopic tubes are called xylem. So that's how water is moved into the leaves of the plant. Light, of course, is provided by the sun. And then when we look at the other side of the equation, our two products, glucose and oxygen. Glucose is produced in the Calvin cycle portion of photosynthesis. We'll learn more about what that means later, but It's the second half of photosynthesis, if you will. And after glucose is produced in the Calvin cycle, often the glucose molecules will be converted into other more complex sugars like sucrose, which will then diffuse into the phloem. Phloem is another type of tubing. It's found just underneath the bark of most plants, and the phloem allows the sugar to be transported from the leaves down the stem all the way down to the roots and to the fruits of the plant, and so that all the parts of the plant receive that food, that sugar that they need for energy. The other main product of photosynthesis, of course, is oxygen. Oxygen is given off as a gas, and much of that oxygen will escape the leaf by passing through the stomata. The same openings that allowed carbon dioxide to come into the leaf are now allowing oxygen to escape the leaf, and so that oxygen is going to end up being released out into the environment. Okay, now let me go back here. When you look at this equation, the overall equation for photosynthesis, scientists have sort of known that this is what happened in photosynthesis for a long time. For a very long time, they did not know how it happened exactly, but they knew that somehow plants were taking in carbon dioxide, water, and light, and that somehow glucose and oxygen were being produced. The oxygen turns out to be a big mystery. For a long time, it was a mystery where the oxygen released by plants comes from. Does it come from the splitting of carbon dioxide molecules? Because you see, carbon dioxide does contain 2 atoms of oxygen. Or does the oxygen come from the splitting of water? In other words, the oxygen that a plant releases, is it this oxygen here? Or is it this oxygen? Or maybe it's a combination of both. And this was a big mystery for quite a while. In the 1930s, a scientist by the name of C.B. Van Niel, who had been working with photosynthetic bacteria, he was the first to sort of adamantly suggest that the oxygen released in photosynthesis does not come from splitting carbon dioxide, but rather it comes from the splitting of water. And he had observed some things in bacteria that led him to believe this. So this wasn't just a guess, it was a very well-informed hypothesis based on observations he had made in bacteria, photosynthetic bacteria. But he did not have absolute proof. The proof would come about 20 years later, when the technology finally arrived that allowed scientists to use radioactive tracing, the ability to trace the movement of radioactive isotopes. And so I'll explain that a little bit. So in the 1950s, this is about 20 years after Van Niel proposed the water splitting hypothesis to explain how plants give off oxygen. In the 1950s, an experiment was done using radioactive isotopes of oxygen. Now, oxygen normally has an atomic number of 8 and an atomic mass of 16, 8 protons, 8 neutrons per oxygen atom. But oxygen 18 has a couple of extra neutrons. So it still has the 8 protons and I guess 10 neutrons. I had to do a little math there. And anyway, it's an isotope. It's a heavier version of the oxygen atom, and it is slightly radioactive. And so you can trace the movement of that radioactive oxygen atom if you have the right instruments that allow you to do this. So basically what these scientists found was that if they watered a plant with radioactive water molecules that had the oxygen 18 form of oxygen, the radioactive oxygen, that those plants would indeed give off radioactive oxygen gas. In other words, the same oxygen atoms that were attached to the water ended up being released as a gas by these plants. But on the other hand, if they fed the radioactive oxygen to the plant in the form of carbon dioxide, those plants did not release radioactive oxygen, but instead that oxygen ended up as part of the glucose. So I know that's maybe a little confusing, but basically what it proves is that every bit of the oxygen gas that's given off by plants when they do photosynthesis, every bit of that oxygen comes from water, and none of it comes from carbon dioxide. The oxygen part of carbon dioxide is not the same oxygen that's released into the atmosphere. So to you and me, this may not be that big of a deal, but it's just a good example of how the great mysteries of biology are often unlocked one little tiny step at a time. This was just the first inkling to how photosynthesis actually operates inside the plant. And of course, it left a lot of unanswered questions that would eventually be answered in the 1960s by a man named Dr. Melvin Calvin. The second half of the photosynthesis reaction is named after him. It's called the Calvin cycle because he is the person who figured out all of the steps of that process. So we'll get to that eventually. Okay, I've already made reference to the fact that photosynthesis occurs in two major steps, or two major stages, I guess you might say. And so I'd like to speak about that just a little bit here. I've broken it down into two columns, and each column represents a major process that occurs during photosynthesis. So the first half of photosynthesis is often referred to as the light reactions, also known as the light-dependent reactions. And there's a very good reason why it's called light-dependent, because as you see, it requires light. This is a series of chemical reactions that must have light as an energy source, and they cannot occur in darkness. Now, this is an interesting phrase. It says light energy is fixed during the light reactions. Well, normally when you hear the word fix, it implies that something is broken and it's being repaired. Of course, that's not what it means in this context. Another meaning of fix is to grab something and put it somewhere where it can't move anymore. To fix something is to sort of grab it and hold on to it and keep it in one place. So plants can do a pretty nifty trick. Plants and other photosynthetic organisms can do something that we animals cannot do. They can take light energy that has traveled from the sun 93 million miles through space, and they can grab that light and hold on to it, or hold on to the energy that's generated by that light, and they can store it and use it later. That's what we mean by light energy being fixed. Water molecules are split. We talked about the proof that this occurs just a moment ago with the radioactive isotopes of oxygen. This splitting of water is called photolysis, and it releases oxygen as a sort of leftover byproduct. So the reason plants release oxygen is because it's necessary to split water molecules during the light reactions, And when you split water, you get hydrogen ions, but you also get oxygen gas. Now, remember, the total purpose, the overall purpose of photosynthesis is to make food, to make a sugar that the plant can use as a food source. None of that sugar is made in the light reactions, but two very important chemicals are made in the light reactions. One of them you know about, ATP. The other you've probably never heard of before. NADPH is similar to NADH, which we learned about in cellular respiration. It's a very similar molecule, except that this has a phosphate group that NADH does not have. But ATP and NADPH are chemicals that store energy. They are energy storage molecules. So the whole point of the light-dependent reactions is to convert light energy into chemical energy. And these two molecules, the ATP and the NADPH, are going to store that energy until it can be used later in the Calvin cycle. Now, in the previous lesson, we learned about the structure of the chloroplast, and I showed you that inside the chloroplast are very tiny disc-shaped pouches called thylakoids, and this is telling you now that that is where the light-dependent reactions occur, inside the thylakoids of the chloroplast. We'll look at that again in just a minute. Let's talk about the Calvin cycle. That's the second half of photosynthesis. Another name for the Calvin cycle is the light-independent reactions, because the Calvin cycle does not require light, at least not directly, as an energy source. It does not get its energy directly from light. The old name for this was the dark reactions, but that's kind of a bad name because that implies that this only happens at night. When you call it the dark reactions, it makes it sound like the light reactions happen during the day and the Calvin cycle happens at night. And that's not true. The Calvin cycle happens mostly during daylight hours, just like the light reactions. But it turns out that the Calvin cycle derives its energy from chemicals, ATP and NADPH, not directly from light. In the Calvin cycle, carbon is fixed. There's that word again. Again, this does not mean that carbon was broken and needed to be repaired. We're talking about invisible molecules of carbon dioxide, which are floating around in the air. And plants have this ability to take that carbon dioxide, grab those carbon atoms, and hold on to them. Use them to make something permanent inside the, well, maybe not permanent, but make something long-lasting that the plant can hold on to and store. And of course, I'm talking about glucose sugar. So when we say carbon is fixed, that just means carbon atoms that are floating around in the air can be taken in by the plant and converted into something useful that the plant can hold on to. Remember, the light reactions produced ATP and NADPH. The Calvin cycle uses ATP and NADPH. The Calvin cycle must have those two chemicals in order to work. So this works really well. The light reactions produces those two chemicals. The Calvin cycle uses them. The main product of the Calvin cycle is actually a 3-carbon sugar called G3P, which you've heard of before because we talked about it when we were learning about glycolysis a couple weeks ago. But eventually G3P is used to make glucose, which can then be used to make starch and cellulose and many other things. But we sort of think of glucose as being the food product of photosynthesis. This is really what photosynthesis is all about, is making glucose that can then be used as food for the plant. And even though the entire process of photosynthesis happens in the chloroplast, the two reactions do happen in slightly different locations. The light reactions take place in the thylakoid of the chloroplast, and the Calvin cycle happens in the stroma. So if you don't remember what I'm talking about, here's a chloroplast, two membranes, outer membrane, inner membrane, and then you have these disc-shaped pouches. That's a thylakoid. So this is where the green chlorophyll is located. This is where the light energy is being trapped. This is where the light-dependent reactions occur. And then out here in the stroma, this fluid-filled space in the middle of the chloroplast, this is where the Calvin cycle is going to be occurring. Okay, I really like this image that's from the textbook. I don't remember if it's from Biology in Focus or maybe it's from the old Campbell, the big Campbell textbook that we used to use, but I really like this because it shows how inside the chloroplast, this big oval is one chloroplast, it shows how the light reactions and the Calvin cycle work together and how all of this fits together. So notice we have light coming in and shining on the green chlorophyll inside these thylakoids. Each little disc is a thylakoid, so this is a whole stack of thylakoids. Do you remember what that's called? An entire stack of thylakoids? That's called a granum. G-R-A-N-U-M, granum. And the light is going to be absorbed by the chlorophyll. Water molecules are going to be split, which releases electrons that can move through the electron transport chain, but it also releases oxygen. So notice oxygen is given off as a product of the light reactions. And if you follow the arrows, if you look at what the arrows are pointing to, the light reactions produce oxygen, but they also produce two other things, NADPH and ATP. Those are the three things that the arrows are pointing to. So ATP, NADPH, and oxygen are the three products of the light reactions. ATP and NADPH are needed over here in the Calvin cycle because these are the two chemicals that are going to supply the energy to make this Calvin cycle go around. What does the Calvin cycle do? It takes carbon out of the air, carbon dioxide, and it produces a sugar. Specifically, it produces G3P, which can then be converted into glucose. What else does the Calvin cycle produce? It produces ADP, which is the stripped down low energy version of ATP. And it also, the Calvin cycle also rips off the hydrogen from NADPH. So NADPH gets oxidized and turns back into NADP+, which can go back over here and be reused in the light reactions. So the Calvin cycle gives off chemicals that can be used in the light reactions. The light reactions gives off these two high energy compounds which are needed in the Calvin cycle. See how it works together? A beautiful system that works wonderfully inside the plant. Okay, you simply cannot fully appreciate how photosynthesis works without understanding a little bit about the nature of light, the physics of light. So light travels from the sun to the earth in waves of light energy. The visible light waves, what we sometimes refer to as white light that shines down on the earth from the sun, is really a mixture of many different wavelengths of light energy. What we perceive when we look at objects, we perceive this as colors of light. These are the colors that our eyes can detect. They also happen to correspond to the colors or wavelengths of light that plants can use for photosynthesis. So this is a good time to mention our extra credit for this week. I've been doing extra credit phrases. This time it's a question. So the first 10 students to email me the answer to this question will receive extra credit for this week. And you just need to put extra credit in the subject line, and then you can answer the question in the body of your email. And it can be a very simple answer. A one-word answer will suffice. Here's the extra credit question. Which color of light is least absorbed by chlorophyll A? Which color of light is least absorbed by chlorophyll A? And you don't need to worry about Googling the answer, because I'm going to tell you the answer in just about 1 minute. All right, so the way pigments work, chlorophyll is a pigment, and the way all pigments work is a pigment is a chemical compound that absorbs some colors of light and reflects others. So if you are wearing a red shirt, maybe you're an NC State fan and you're wearing a Wolfpack red shirt, the pigments, the red pigments in that fabric are absorbing orange light, yellow light, green light, blue light, violet light. But those same red pigments are reflecting red wavelengths of light. The reason we see that shirt as red when we look at it is because red light is hitting the fabric and bouncing off. And that's the color, that's the light that's coming to our eyes when we look at that shirt. So that's what pigments do. That's what all pigments do. And chlorophyll, of course, happens to be a green pigment. Chlorophyll is the reason why leaves are green. And so let's talk about what happens when white light strikes the green chlorophyll of a plant. So first of all, visible light, which is the same thing as white light from the sun, is actually a mixture of different forms of light energy that each have different wavelengths. That's what these numbers are talking about. And we know this is true because we've all seen rainbows. We've all seen what happens when sunlight is diffracted by raindrops in the atmosphere. We can see red, orange, yellow, green, blue, indigo, and violet. So we know that when the sun shines on a plant, all of those colors are striking the chlorophyll. If you've ever learned these colors before, you've probably learned them in reverse order so that it spells out a name. Roy G. Biv is an easy way to remember the colors of the visible spectrum. Notice that there are some slight differences in terms of not only wavelength, but also energy. So the further you go to the left towards violet, toward the violet end of the spectrum, that color of light actually has slightly higher energy level than, say, red light, which is over on the right-hand side. So the further we go towards red, the wavelengths get slightly longer, but there's actually slightly less energy available. That turns out not to be terribly significant when it comes to photosynthesis, because photosynthesis is really more about which of those colors are being absorbed by the chlorophyll and which ones are not. So this is kind of a really nifty way to understand this. You've got white light from the sun shining down on the green chlorophyll, and notice most of the colors, the violet and the blue and the a little bit of the yellow and even the red and the orange, those colors are being absorbed, and that's providing the energy for photosynthesis. But the green light is hitting the chlorophyll and either bouncing right off, that's reflected light, or it's just passing right through the chlorophyll. That's called transmitted light. So green light is not absorbed by chlorophyll. Chlorophyll A is the most common form of chlorophyll. It's the green pigment. And when all the colors of light strike the chlorophyll, the color that is least absorbed is the green light, and the other colors are absorbed. So this is a graph. These graphs they have a lot going on. It's a little confusing, but I just want to call your attention to a couple of places here. If you look at this middle graph, it shows the rate of photosynthesis when different colors of light are shining on a plant. So if we shine violet light on the plant, and only violet light, the rate of photosynthesis is fairly high. If we shine red-orange light on the plant, the rate of photosynthesis remains high. But if we shine only green light and yellowish green light on the plant, notice the rate of photosynthesis drops way, way, way down. Well, that's not surprising, because remember what happens. Green light is either bouncing off of the chlorophyll or passing right through the chlorophyll. The green light is not absorbed by the chlorophyll. And so, of course, the rate of photosynthesis is going to drop. Now, you also notice the rate does not drop all the way to zero. Photosynthesis does not completely stop when you shine green light on a plant. And that is because chlorophyll A, which is the most important pigment for photosynthesis, chlorophyll A is not the only pigment that most plants have. Many plants have a second type of chlorophyll called chlorophyll B, and most plants have carotenoid pigments, red, orange, and yellow pigments, and they are able to absorb some of the energy from green light. So chlorophyll A might not be able to absorb green light, but carotenoid pigments can absorb some of that green light energy and then transfer that energy over to the chlorophyll so that none of that light energy gets wasted. So this is another way of kind of driving home this point. This is showing the inner workings of a machine called a spectrophotometer, which measures how much light is able to pass through a sample. And you can see that the sample that they are using here is chlorophyll. That's the green pigment that has been extracted from a leaf. And if you shine green light onto that sample, it passes right through. Notice the needle is way over to the right. That's a really high percent transmittance. It shows me that hardly any of that green light was absorbed by the green chlorophyll. It just passed right through. On the other hand, if we change the color of the light beam to blue light, much of that blue light energy is going to be absorbed by the chlorophyll. It's not going to pass through the sample, and therefore it's not going to strike this sensor. This is a light sensor. Notice the needle is way over to the left, showing that very, very little of that blue light was transmitted through the chlorophyll. Why is that? Because chlorophyll absorbs that blue light energy, but chlorophyll is unable to absorb the green light. So again, back to the extra credit, it is green light that is least absorbed by chlorophyll A. So the pigments that make photosynthesis possible, there are several of them. The primary pigment is chlorophyll A. You can see right here it's a magnesium-containing pigment molecule, very complex structure. It's sort of a dark green color, and this is what gives the leaves of most plants their green color. Some plants also have a slightly different version of chlorophyll called chlorophyll B. It's a pale olive green color, and many plants have what are called accessory pigments or helper pigments, which are sometimes also referred to as carotenoid pigments, and those are red, yellow, and orange. And we start to see those pigments in the fall. In autumn, when the green chlorophyll is no longer being produced, we start to see some of those red, orange, and yellow colors, which have really been there the whole time, but we only see them in autumn when the production of chlorophyll A stops. Okay, I'd like to finish our lesson today by walking you through the steps of the light-dependent reactions of photosynthesis. Remember, this is the first half of photosynthesis. This is what happens within the thylakoids inside the chloroplast. So let's look here. This is called photosystem 2. A photosystem is just a collection of pigment molecules, mostly chlorophyll A molecules, but maybe chlorophyll B and maybe even some accessory pigments, orange and red pigments. It's pigment molecules and also some proteins that are going to pick up any excited electrons that leave the chlorophyll. The photosystems are embedded in the membrane of the thylakoid. So you'll notice right in the middle of photosystem 2 is a very important molecule of chlorophyll A called P680. This is called the reaction center. And basically all of the energy that's absorbed by all of these chlorophyll molecules is going to funnel down to this one molecule in the center. And there's going to be enough energy to excite electrons in this molecule of chlorophyll until they have so much energy that they leap out of the chlorophyll and they get grabbed up by a protein called the primary acceptor. So let's go back to the beginning. First we have light striking the chlorophyll, and that light energy is absorbed, at least most of it is, not the green wavelengths, but all the other colors, especially red and orange and blue and indigo and violet, those colors are absorbed by the chlorophyll, and all that energy gets funneled into this central molecule called the reaction center until it has so much energy from that light that electrons within the chlorophyll become excited. And they have so much energy now that they leap out of the chlorophyll and they got to go somewhere, so they get picked up by a protein called the primary acceptor. So those electrons are leaving photosystem 2. That means photosystem 2 has just been oxidized, right? It has just lost a couple of electrons. Well, those have to be replaced. And the electrons, in order to replace them, a molecule of water, here's water, has to be split. Now, when water is split, that produces three things. It produces 2 hydrogen ions. It produces 1 oxygen atom. They're calling it 1/2 O2, but what is that? That's just O, right? So an oxygen, which will eventually find another oxygen and form an O2 gas molecule, which will eventually diffuse out and into the atmosphere. So this is the oxygen that's given off as a gas. And then the third thing that's produced is 2 free electrons. So splitting water, which is called photolysis, produces 2 hydrogen ions, oxygen, and it also releases 2 electrons. And those 2 electrons replace the 2 that were just lost from photosystem 2. Remember, when light hit the photosystem, it excited 2 electrons. They had so much energy that they leapt out of the chlorophyll and they moved over to this protein called the primary electron acceptor. Now, where do those 2 high energy electrons go next? Well, they travel down an electron transport chain. We've seen this before. We had an electron transport chain in the mitochondrion, on the inner membrane of the mitochondrion. And it had some of the same proteins, like cytochrome proteins. And you know what an electron transport chain does. It transfers electrons from protein to protein to protein to protein until they eventually reach some other destination. And in this case, these high energy electrons, they gradually lose their energy as they pass down the electron transport chain. And that flow of high energy electrons creates a proton gradient, just like it did in the mitochondria. And we're going to have eventually chemiosmosis, just like we did in the mitochondria. What's that going to do? It's going to produce a lot of ATP using the ATP synthase protein. So we'll get to that. I'll come back to that. But basically what has happened so far, light energy has excited a couple of electrons. They have left photosystem 2. They have passed down the electron transport chain. And now those same two high energy electrons are going to reach their destination, which is photosystem 1. Now you might be thinking, what the heck, Mr. Kapnis? Why is this photosystem 1? Shouldn't this one be named photosystem 1 and this one be named photosystem 2? And the reason they're named backwards is just because of the order in which they were discovered. Photosystem 1 was discovered first, and therefore it was named number 1. So I know it's backwards. The first photosystem to receive light energy is photosystem 2, and the second photosystem to be energized by light is photosystem 1. Okay, so you just have to get used to the fact that they're numbered backwards. When light strikes photosystem 1, same thing, all these chlorophyll molecules are absorbing that light energy. They're funneling that energy to the middle of the photosystem. This special molecule called P700 is the reaction center of photosystem 1. And it's going to have so much energy being funneled into it that once again, 2 electrons are going to be re-energized and re-excited, and they're now going to once again have so much energy that they're going to leap out of the chlorophyll. There's those 2 high energy electrons. They've been juiced up with light energy, and now they're going to leap out of the chlorophyll, and they're going to be picked up once again by a primary acceptor protein. And once again, those high energy electrons are going to travel down an electron transport chain. Not the same one, not this electron transport chain, but a different electron transport chain. And this time, at the end of the electron transport chain, there's a special protein called NADP reductase. It is going to pick up those 2 high energy electrons, and it's going to pick up a couple of hydrogen ions, and it's going to stick those onto a molecule called NADP+. So this enzyme, NADP reductase, that's the name of the enzyme, its job is to reduce NADP+ by adding the 2 high energy electrons to it. And when NADP+ is reduced, it picks up the 2 electrons and it becomes NADPH. So, by the end of the light-dependent reactions, light energy has been absorbed twice, once here and once here, and 3 things have been produced. Make sure you know the 3 products of the light-dependent reactions. They are oxygen, ATP, and NADPH. And of those 3, the 2 that are needed in the Calvin cycle are going to be ATP and NADPH. Okay, let's take a closer look at how the ATP is made, how these ATP molecules are made in the light-dependent reactions. So we are looking inside one little disc, one thylakoid. This is a thylakoid. Notice it is surrounded by a phospholipid bilayer membrane. Here is light striking photosystem 2. What does that do? It excites electrons. Where do those electrons go? They go to the primary acceptor, and then they get passed to the next protein, which passes them to the next protein, which passes them to the next protein. Remember, this is called the electron transport chain. And where do those electrons end up? Over here at photosystem 1. So light has caused high energy electrons to leave photosystem 2, pass down the electron transport chain, and go all the way to photosystem 1. That high energy flow of electrons creates enough energy to pump protons across the thylakoid membrane and into this space, which is called the thylakoid space. Sometimes it's called the lumen, L-U-M-E-N. That's another name for it. So what happened was 2 protons from the stroma out here, those 2 protons have no choice. They are pumped into the thylakoid space. And every time light hits photosystem 2, every time electrons start moving down the electron transport chain, more and more protons are being pumped into the thylakoid space. Well, once they get in here, they're trapped, right? Because they can't cross a phospholipid bilayer. So the protons build up inside the thylakoid space. The pH level starts to drop, because the higher the concentration of protons, the lower the pH. It actually becomes acidic in here. Also, this is a voltage situation, right? Because you got all these positive charges. So this is like a positive electrical charge on the inside. It's a slightly negative electrical charge out here. There's all kinds of things going on. But the most important thing to notice is this is a really, really high concentration of protons, because the electron transport chain has forced protons to be pumped into this thylakoid space. We have a high concentration of protons in the thylakoid space. We have a lower concentration of protons in the stroma. That is called a proton gradient. It's also called a proton motive force. And it's important, because when this valve opens up, this is ATP synthase, you know what's going to happen. Those protons are going to shoot across. They're going to move through ATP synthase with a great deal of energy. Enough energy that every time a proton shoots across, that little rotor inside ATP synthase spins around, and it adds a phosphate onto ADP. What does that make? You guessed it, ATP. So ATP is made in the thylakoid by the process of chemiosmosis. Remember what chemiosmosis means. It's the diffusion of protons through ATP synthase. It's the high energy flow of protons that spins a rotor inside the ATP synthase protein, and every time that rotor spins around, ADP gets phosphorylated to make ATP. And so, plants have 2 ways to make ATP, right? They make ATP in aerobic cellular respiration, which is happening in the mitochondrion. But now here we are in the chloroplast, and the plant is using light energy to generate ATP through the process of chemiosmosis. And of course, these ATPs are going to be used for one designated purpose, and that is to supply energy for the Calvin cycle. Now, let's not forget about our other product of the light-dependent reactions. Remember, light hits photosystem 1. What does that do? It re-excites those same electrons, the ones that were originally over here. Now those electrons get re-excited in photosystem 1, and the high-energy electrons travel to NADP reductase, and they get added on to an NADP+, which makes NADPH. ATP and NADPH are the two high energy molecules which are required by the Calvin cycle. They're going to be used up in the Calvin cycle. The other thing you don't want to forget is the importance of water. Why do plants need water for photosynthesis? Because every time light hits the chlorophyll, electrons are lost and they're never coming back again. This is a one-way trip from photosystem 2 to photosystem 1, and eventually where do those electrons end up? They end up right here in NADPH. So this chlorophyll in photosystem 2 would constantly be running out of electrons. They have to be replaced. How does the plant replace those lost electrons? By splitting water. Remember, that happens in photosystem 2. Light energy is used to split water. That process is called photolysis, and that happens in order to release electrons to replace the electrons that were lost from photosystem 2. So once again, I've drawn this out. This has now become a very long lesson, and I apologize for that, but I had a lot to cover. And so we will tackle the Calvin cycle in our next lesson. Until then, I hope you do well, and I hope you're studying and working hard, and I will see you soon.